Hello everybody, and thank you for joining this webinar on COVID-19 search strategies in PharmaPendium, where we will focus today on DMPK data and predicting drug-drug interactions. My name is Marnix Wieffer, and I'm Senior Solution Marketing Manager Drug Safety at Elsevier, and I'm joined today by Thomas, who is a PharmaPendium Product Manager. A few housekeeping comments before we really kick it off. First, we would recommend to watch this presentation in full screen mode, and please make sure, of course, that your audio is turned on. Next, during this webinar, you have the possibility to ask questions by typing these into the Ask a Question box. We will address any questions at the end of the webinar. Also, after the webinar, if you have any additional questions, or if you feel that your questions have not been fully answered, always feel free to send me or Thomas an email or send us a message on LinkedIn as well. Also, you can access the various links that we've attached to this presentation through the attachment section that you will find below the viewing screen. I didn't manage to attach the slides yet, but after this presentation, I will also upload the slides under the attachment, so you're able to access those slides in a couple of hours after the ending of this presentation. Now last, if you'd like us to follow up with you based on what you've learned during this presentation, maybe you have further questions, or you would like to learn more about the solutions that we're currently presenting, like PharmaPendium, and how they can help you to uncover critical DMPK data and predict and anticipate DDI risk. At the end of the webinar, we'll put up a little vote, and if you would like us to follow up with you, simply select yes. If you don't want us to follow up with you, you don't have to select anything, or feel free also to select no, of course. Now, for today, there's a couple of things we would like to discuss with you. First, we're going to provide a brief introduction into how Elsevier is supporting the fight against COVID-19, why PharmaPendium is a critical resource for repurposing drugs, supporting the fight against COVID-19, and then we're going to dive into a couple of DMPK and DDI specific workflow examples. So first, Thomas is going to show us how to uncover data that will elucidate how patient complications and patient background can influence therapeutic PK profiles, and how that again can influence your dosing strategy on COVID-19 patients. Then we're going to explore what drugs for the treatment of COVID-19 symptoms have favorable administration routes. Let's assume a patient has limited routes of administration available. How can we identify the right drug for the right route of administration using PharmaPendium? And last, what DDIs can I expect now in creating various uncommon drug combinations as we're testing approved but still new drugs in patients that haven't been frequently exposed to these drugs before, and hopefully there's still some time for Q&A. I hope at the end of this meeting, hopefully you have some new insights into how DMPK data and DDI prediction in PharmaPendium can help with the repurposing of drugs for the treatment of COVID-19 infections and COVID-19 symptoms. And for those on the call already using PharmaPendium, hopefully you got some practical insights into finding DMPK data and mitigating DDI risk using PharmaPendium. Now with that, for this audience, COVID-19 doesn't really need much of an introduction. Of course, everybody's fully aware of the worldwide pandemic we're currently in. And this pandemic is, of course, far from under control. And as the pandemic is progressing, there will be continuous need of novel medication to treat COVID-19 and COVID-19 symptoms. At this point, we have close to 2 million new cases every week and over 35,000 deaths every week. And of course, next to the severe healthcare consequences and the overloading of the healthcare systems worldwide, there's of course a major impact on world economy as well. Now, not surprisingly, Elsevier, as data and analytics company with a very strong focus on pharmaceutical R&D, is of course well positioned to support the fight against COVID-19. And therefore, we've made many data and documents freely available in the Corona Research Information Center. And as mentioned, I provide the link to this Corona Research Information Center in the attachment section of this presentation, so you can find more information there. Further, if you're an R&D professional working on COVID-19 related projects, you can apply for access to Elsevier's Coronavirus Research Hub. And through this research hub, you'll get access to over 15 Elsevier solutions. And those solutions include, for example, Science Direct, so Elsevier's literature platform, as well as, for example, Reaccess, as well as PharmaPendium. And about PharmaPendium, of course, you'll learn more today. You will find these links to these resources again by clicking on attachments under your video player. Now, there's been quite a bit of news around vaccines that are currently looking quite promising, that are currently in the pipeline. But next to those vaccines, there are also many existing drugs that are being tested for the prevention and treatment of COVID-19 and COVID-19 symptoms. And this is, of course, a very sensible strategy, as developing a totally new drug can take up to 10 years and more and really consume significant amount of budget and resources. There's, of course, a couple of numbers going around, but estimates range from developing a new drug can cost between 300 million and $2.5 billion US. And if you, of course, can work with an already existing drug that already went through a regulatory approval, that can save significant time and resources. Now, now to maximize such repurposing and treatment success of an already approved drugs, you really need to be fully informed on all regulatory data available on those drugs that are already on the market. Now, regulatory approval documents are, of course, a crucial source of precedent data, for example, safety data, DMPK data, as well as efficacy information, as these data that are inside these regulatory documents have already been evaluated, commented on, and approved of by regulatory authorities. So that's the highest grade data that you can get your hands on to further drive your drug development and approval. Now, most on the call know that finding information in regulatory documents is really challenging. It's really hard to know where you should be looking for and what information is available. And as many of you are aware, many FDA approval documents are scanned image files and cannot be searched in full text. And without reading the full document, or maybe for some questions you need basically to read all available approval documents, you would never know if the document contains the relevant information. Now, to address this issue, we have developed PharmaPendium, and that really aims to help you to overcome these bottlenecks and unlock critical regulatory data on approved drugs. Now, how do we do this? So basically what we do, we take EMA and FDA approval documents, and on top of that, we also take FDA advisory committee meetings, books, as well as journal articles. Then, especially those regulatory documents, we really turn into full text searchable content. So what used to be often a scanned image file, it can now be searched in full text thanks to PharmaPendium. And then a panel of medical experts reads all the documents, so including the approval documents, in this case also the literature, and extracts out drug safety data, DMPK data, as well as efficacy data. And then we make those data discoverable by mapping the data to expert biomedical taxonomies. Many of these are in-house developed and exclusively available within PharmaPendium. And a couple of those taxonomies that we're going to show you today are PharmaPendium's Pharmacokinetic taxonomy, as well as PharmaPendium's enzyme and transporter taxonomy. Now, next, we overlay this database with an intuitive user interface and make results accessible through this UI. However, there's also other ways of working with the data, as we also allow, of course, data export, as well as API access, whereby you can bypass the user interface and directly access the PharmaPendium data. Now, in conclusion, what we deliver is then a searchable index database where every data point that we extract is linked back to the original document, and where the original document is also accessible. All extracted information is searchable across drug class, but also based on indication, as well as primary targets as well. And the data can be used into analytical tools and in-depth data analysis. And thereby, PharmaPendium really delivers crucial information for the most comprehensive risk-benefit assessment during repurposing of already approved drugs. Now, with that was kind of my introduction into PharmaPendium, and I would suggest Thomas to take over. So activate screen share so you can start presenting your example.
Thank you, Marnix. Do you see my screen now?
I think we still look at your. Yeah, now we see it. Now we see the slide.
Great. So last time, during the last webinar a week ago, I talked about the case of the repurposed drug Tofacitinib, which is tested for clinical trials to help fight the COVID situation, and what adverse effects you can expect at therapeutic dose, but also if you need to increase the dose. Today, actually, I'm going to show you about when it comes to repurposed drugs, if some population can be a concern when you are going into clinical trials, because dose exposure can drastically change for the same dose from a patient to another. So what drugs should be avoided, or how should dosing be adjusted? And what information on patient stratification I can get and how it can impact clinical trial design. Now, I came across a very nice article about some design flows for some early first wave COVID-19 clinical trials. But I will go, at the end of this presentation, I will go back and show you about this use case in PharmaPendium. So for this case, I will go to PharmaPendium, and I will start the search here on browse drugs. And I will put again Tofacitinib. But now I want to have a more comprehensive starting point. So I will broaden my use case to all the drug class where Tofacitinib belongs to. And this is the disease-modifying antirheumatic drug, DMAD. And this is very easy to do in PharmaPendium. You just click here above the drug category, and I just populated all the approved drugs for the disease-modifying antirheumatic drug, DMAD. And I can click on View All to have all the list of these drugs. Now, I want to pay your attention on this list, actually. You have, like me, followed the news over the last 6 months, and you see in this list some drugs that should sound familiar. So you have chloroquine, you have hydroxychloroquine, you have sarilumab, of course you have the baricitinib, you have azathioprine, tocilizumab, and so on. So, as you know, this drug family got a particular attention this year for repurposing studies. You see all the manually extracted data our expert team performed for this drug. So to understand if for the same dose, a different exposure can be expected or not for different population, I will check on the PK data module here. And you want to know that because if you have a population with renal or hepatic impairment or even ethnicity differences, and change that with the broad spectrum of COVID patients profile, these possible at-risk groups may occur for clinical trials. So I will open this link. So what you see here is a very nice table where the pharmacokinetic parameters were extracted and reported along the drug of interest. The species, the study group, the dose, the route, the parameters, and so on. Now, for drug exposure insights, what is very nice in PharmaPendium, I can filter by AUC and Cmax to understand indeed if a drug regimen needs or should be adapted. So I will add in yes. So I filter for Cmax and AUC, and now I have still like more than 6,000 records. So for like large number of records, I like to go straight to the export file and have filters so I can get the information I need quick. So I will press on export. But what I did for the purpose of this presentation, I've already prepared this file where I just put some more like filters. And now you can see here, for example, you have exactly the filters and the table results that you have for PharmaPendium that I just showed you. So, and what I like to do is I can quickly scan through the study groups by checking the filters like this. And then I can quickly scan about like what the study group were studied in this clinical trial. So I can have a precise idea of what you want to look. This is a very nice to have this overview. So for example, for this study group, I want to know a bit more about renal impairment. So this is easy. I will just like put renal. And now you can see the study group with like mild renal impairment, end-stage renal disease, and so on. Everything related to renal impairment I can quickly access. And as you can see here again, you see the Tofacitinib and some other drugs that you can even like filter furthermore. So for example, now you can go to the source of documents to see a bit more about contextual description about what's going on with this study group. So what I will do, I will just like click on this link. And this is what I have. So from the source document, you have this table with different type of renal impairment cohorts. Now you have the PK parameters with Cmax, we have the Tmax, you have the half-life, AUC, different clearance, and so on. Now you can quickly see from the healthy cohort you have here to the mild, to the moderate, to the severe cohort, and so on. How does the Tmax, the AUC, and the half-life evolve? You have the dose, you have the amount of patients tested, and this is very nice to see. So for example, for half-life, you can see that from the healthy cohort, this half-life almost doubled for half of the dose from the healthy cohort. So this could be an indication of how you can adjust the dose regimen according to the dose exposure and so on. And this is where all our experts, MDs and PhDs, extract all this data, manually extract this data, process them, and put them back in PharmaPendium so you can quickly, like I did earlier on, just filter on these parameters. Now, what this information tells me, what can I see from the other page? I have like also some description about Cmax decrease with increasing body weight. However, the effect of body weight on Tofacitinib PK is not considered clinically relevant. You can also have some information on gender and race, American versus Japanese, very interesting. Sometimes different ethnicity can react differently from doses. But what it says here, genders and race were shown to have an effect on PK of Tofacitinib, but were considered not to be clinically relevant. Again, maybe not a blocker, but you still want to have a close monitor should you have different ethnicity. Now, what I see here is something quite interesting. I see like no clinical studies with Tofacitinib were performed in patients with severe hepatic impairment. So maybe you can take the choice to remove patients with severe hepatic impairment because it could be that your drug is not recommended. But for some reason, let's say you need to perform clinical trials on severe hepatic impairment. So in PharmaPendium, what you can do, you can quickly go back to the page that you come up from. And what I will do, I will just you can clear all the data. And basically you can, again, export to have a better comprehensive view of what eventually I can see and what was the strategy around hepatic impairment. So for the sake of the presentation, I did the same. I will export, and now I have this already prepared Excel file. So what I can do from this one, again, I can quickly dive into this filter here. Oops. Here, and I will just put hepatic. And now again, you have all the study group with some hepatic dysfunction, severe hepatic impairment, moderate hepatic impairment, and so on. You have the drugs. You can also filter by drugs, and so on. You have the value, the parameter. Now you can see quickly here that maybe for some severe hepatic impairment for all the drugs, you don't have a dose related. So again, this could be an indication that this drug was not recommended for severe hepatic impairment. But like I say, sometimes you need to add this drug to severe hepatic impairment for the clinical trials. So again, what you can see from PharmaPendium, from very quickly results in this Excel spreadsheet, you can see some dose with mild hepatic impairment, moderate hepatic impairment, and so on with a different dose. And again, you can dive into the filter and see what are the dose limitations. Again, you can also apply the same strategy that I did for renal impairment and dive into the source itself. But I can already have crucial information on this page. You can see that the maximum dose, even for moderate or mild hepatic impairment, was 10 milligrams. And then the lower dose was 3 milligrams. So this could give you an indication to where to start for severe hepatic impairment patients in clinical trials. So maybe you want to start even lower than 3 milligrams. But this is what PharmaPendium can give you in access very quickly. Now, I want to go back to the document that I landed from renal impairment. So you have the PK parameters, you have a bit of population indication, and I want to pay your attention on this table here. Now you have the different phases for these drugs what was tested, clinical phase 1, phase 2, you have a bit of the different studies, and importantly, you have the cohort by age. You know how important for the COVID-19 different cohort by age, since mainly the COVID-19 patients and aggravation of this condition is for age 65. So that will give you a good incentive of how many patients were tested for these drugs to be approved. And you can see, for example, phase 1, you have 557, phase 2, 571, and for phase 3, you have more than 3,000 patients tested at under 65, but below 65, you had also like more than 550 patients. Now, why this information is quite important? Because I can go back now to my previous slide about the article I found very interesting for this early COVID trial design. Some flaws were spotted. And you can see here, for example, nearly one half were designed to enroll fewer than 100 patients. So you can see a bit now the difference between what I just showed you in terms of volume of patients. And maybe you don't need to go that high level of or high volume of patients. But you can see that maybe fewer than patients limiting their usefulness to assess modestly sized treatment benefits may be a problem down the line. And again, here it says, given the urgency of identifying definitive evidence on potential COVID-19 treatments, this is an instance where we wish we didn't have to say further research is needed because of basic trial design shortcomings and small trials. So again, I just want to say that in PharmaPendium, we have different modules with different use cases. And this use case is better addressed maybe for efficacy module in PharmaPendium, and this could be the case of a next webinar that we can prepare with Marnix about efficacy module, who can give you critical information on how to study, how to design clinical trials to have success, to have the better success for your drug repurpose. And that's it for me, Marnix.
Okay, thanks. Let me start sharing my screen. So give me a couple of minutes to get this started. Screen share, 2 screens, next. And I would like to share this screen. And okay, this should work now. And let me open up my presentation. Let me go here. Really to further support, because to really thank you for that interesting workflow example. And I think it touched upon a nice aspect of PharmaPendium, that the data quality in PharmaPendium is really high and is really kind of can be readily used in analytical modeling, pharmacokinetic modeling approaches, and really to further support the fight against COVID-19 and really to kind of make use of this data quality. We started a collaboration with a French pharmacokinetic analytics company, which is called ExactCure. And maybe in a nutshell, what ExactCure is doing, they are modeling the PK behavior of a drug at individual patient level. And these models, of course, need the highest quality of data. And really that data is now supplied by the PharmaPendium team. So in a nutshell, Elsevier and ExactCure are working together to build personalized drug-specific exposure models that allow the prediction of pharmacokinetic properties of the repurposed drug at individual patient level. And as Thomas just mentioned, right, it can be that a patient is suffering from renal impairment, and this would need, therefore you would need to adjust the dosing of your drug to your patients. And together with ExactCure, we aim to make that possible. If you think that's interesting and would like to learn more, on the 21st of October, we are hosting a webinar together, optimized personalized drug dosing for patients with drug-specific pharmacokinetic data models. You'll find the link to this webinar, so how we can use data and modeling to optimize dosing for specific patient populations in the attachment section. Now let's dive into the next example that we prepared. Just keeping an eye on the time here. Maybe I need to cut down one example. Let's say the question could be, what drugs for treatment of COVID-19 patients have favorable administration routes? So you can imagine that for patients at intensive care, all intravenous routes have been exploited, and maybe oral administration is also not possible. And maybe we would need to apply an antibacterial drug. And we maybe think, well, could we maybe use inhalation? And if we use inhalation, what are the effects then on area under the curve? So let me then dive into PharmaPendium and show you how we can address this question. So hopefully you can see my screen. So this would be a question we can really address using the pharmacokinetic data module in PharmaPendium. And as Thomas already showed as well, we can filter on different, we can limit our search based on different drug taxonomies, including drug class or drug name, drug by primary target, or drug by indication. And in this case, we are interested specifically in the indication bacterial infectious disorder. If we click on Add Drug by Indication, this will open up PharmaPendium's indication taxonomy, which will allow us to look for all the drugs that have been tested against, based on the data that's in PharmaPendium, against bacterial infectious disorder. If we open up this taxonomy, we can dig even deeper. Now, what we can do here as well, maybe we're only interested in drugs that have been approved to act on bacterial infectious disorder. To this end, we can open our sources filter and say I'm only interested in the drugs where the drug association against this indication was found in the FDA label or the EMA annex. And we click on Apply. And we click on Done. And click on Search. So this gives us over 80,000 records of PK data lines on drugs approved for treating bacterial infectious disorder. Now let me quickly zoom out a bit and close this. And we really extract a lot of data, as already shown, species, study group, dose, route, parameter, the parameter value, and of course the links to the original source documents as well. And in this case, maybe we're only interested in the PK data available on the route of administration inhalation. So thanks to PharmaPendium's intuitive filters, we can simply go to routes of administration, and we get an overview of all the routes of administration associated with these data lines, and we can simply select inhalation and click on apply, where we trim it down to 1046 records. Now, next, if you would like to know how a certain dose of inhalation affects the area under the curve, we can simply filter on the parameter range, where we can then open up PharmaPendium's filter for parameter range, which is based on PharmaPendium's pharmacokinetic data taxonomy. We can either look for the term of interest in any of the upper level categories, or we simply type area under the curve and select area under the curve data. So now we really effectively trimmed it down to 240 data lines. And what we can see is that that within just a few clicks, we really kind of trimmed down our data to 5 drugs, which we can show through Show Drugs In, that are mentioned here, that have been approved as antibacterial drug, that for which we have data available on area under the curve as displayed over here. Now this is of course really nice, because this gives you the opportunity to compare the dose that you apply versus the result of the area under the curve. So you can, for example, also use this approach to compare what a dose of inhalation versus a dose of let's say a pill, how this would influence the area under the curve, just to make sure that you can dose this drug through your most appropriate route of administration at a safe level. Now, of course, from this point, we can simply export this data through our export option. But again, we'd like to also mention that you can access PharmaPendium data through an API, an programmable interface, which will allow you to bypass the PharmaPendium user interface and directly access the data from within PharmaPendium. And this is, of course, a great starting point for further investigation, where, for example, you can now go and explore on any of these 5 drugs the efficacy data to see what the effect of this dose with this inhalation route, with this area under the curve, is on the clinical outcome for your patients, to further maybe trim down this drug further. Now, let me go then quickly to the next example, keeping an eye on the time, because we also would like to quickly discuss drug-drug interactions with you. Now, people vulnerable to COVID-19, of course, have multiple other conditions and are often already on multiple medications already. COVID-19, of course, especially severe COVID-19 cases are, of course, often happening in the elderly population that often already suffer from obesity, asthma, and various lung conditions, diabetes, heart disease, kidney and liver diseases as well. And it's very likely that they take a mix of multiple medications already before they end up on the intensive care and might need treatment with further medication to fight the COVID-19 infection as well. Now, what we know is that the risk of adverse drug reactions, so where drugs would basically act indirectly on each other's activity through the activity on metabolic enzymes, this risk really increases very significantly when patients are on 4 or more medications. And basically 13% of adults experience potential serious DDIs, which again correlates with the increase in polypharmacy. And what you can see in this graph as well is that there is a significant part of the population, over 5%, and this is still increasing, which is on 10 or more drugs. And now we have to add additional drugs that we're trying to repurpose to fight COVID-19 to this mixture and avoid, of course, severe drug-drug interactions. So a question that we could ask is what enzymes contribute to Tofacitinib metabolism based on fraction metabolized data. Now, depending on your expert level in drug-drug interactions, let me just briefly give you some background onto this question. So first, what is Tofacitinib? Now, Tofacitinib is a drug that suppresses inflammatory signaling, and that could help to suppress a cytokine storm, which is basically the immune system going in overdrive, often seen in COVID-19 patients. And currently this drug is in phase 2 clinical trials. Now, what's then FME, and why is this important for drug-drug interactions? As an example, your drug of interest is maybe processed by the enzyme at the liver, CYP2D6, and it is approximately 90% processed by this enzyme. And this means that the fraction metabolized for your drug by this enzyme is 0.9. Now, if we know from literature and regulatory documents that we know what marketed drugs inhibit or activate CYP2D6, thereby this will enable you to predict what marketed drugs potentially interact with your drug of interest. So this is kind of the logic, and this is of course why FME data is so very important to interrogate DDI risk. However, FME data, as with other drug metabolism data, is often not reported in the literature. And for a detailed description of metabolism of marketed drugs, we really critically rely on what's reported in the full text of regulatory documents. And as mentioned, that's really what we do in PharmaPendium. We read the full text of that regulatory document for you, not just the label, and extract out all the data, including metabolizing enzyme and transporter data. So to address this question, let's dive into PharmaPendium, but not the PharmaPendium the PharmaPendium Pharmacokinetic module, but let's aim for the, now let me find PharmaPendium back again, but let's aim for the PharmaPendium metabolizing enzyme and transporter module, which we'll find over here. Now here we can really limit our search similarly in a similar way as we've done for all the other modules. We can limit our search based on drug class or drug name, based on indication, by primary target, and much more. But in this case, we're specifically interested in a particular drug. So then we can go to add drug by drug or by drug name or drug class, simply type the drug name of interest, and we click on apply. Additionally, on the right-hand side, we can filter our search based on where the data was extracted from. So that's sources, in which species the data was generated, on what enzyme or transporter the data was collected. And here we'll maybe dive in a bit more detail later. We can get a full overview of PharmaPendium's enzyme and transporter taxonomy, which will allow you to search at the enzyme or enzyme class of interest. And we can search by data type. In this case, we're interested in fraction metabolite data. So we simply type FM and select the categories FME and FME in vitro and click on done. If we now click on search, what we get are 9 data lines where we have FME data on Tofacitinib. And what we can see, and let me pin the columns for you to get a bit of a nice overview, and we can do it by simply clicking on this little box here, and we click on pin, and we go to value and click on pin. And if we now look through the data, so what we can see with these 9 data lines where we have a lot of data available, including enzyme and transporter, the value on FME and, for example, what the route of administration, substance, we have experimental conditions, time, the reaction degree, route, dosing. We have a lot of data available that we extract out of these documents. But for this particular example, this is the most relevant data, where we can see the FME value for the different transporters different enzymes available for the drug Tofacitinib. And what we can see is that in this case, CYP3A4 has a FME value of 0.7. In this example, this data was collected in microsomes, as we can see here, liver microsomes, so that's high quality data. And what we can see is that the other enzymes are relatively less responsible for the breakdown of this drug, like CYP2C9 and CYP2C19 as well, because they have lower values. So this is a very nice way to get a quick overview of which enzymes are mostly responsible for the breakdown of this drug. And if you want from here, you can always dive into the original regulatory document. And this, for example, let's dive into the FMA, let me go and look where it is. Let's dive into the FDA approval package document and see what data we got. And what we can do, for example, is look at the original source data, or we can further dive in and look, for example, for summary to look for regulatory comments here and what the sponsor is actually claiming here. And the sponsor recommends dose adjustment for patients receiving drugs that inhibit both CYP3A4 and CYP219. However, Tofacitinib dose adjustment is not warranted when co-administered with a CYP219 inhibitor. So we really get this really detailed insight in dosing and dosing strategy in the context of drug-drug interaction risk. So really through PharmaPendium's expert extracted MET data, manual extraction from regulatory documents and literature, expert taxonomies, and easy access to source documents, we can really identify DDI risk. And these data suggest that there is a high risk to Tofacitinib interacting with CYP3 and 4 inhibitors or inducers. Now, we actually prepared another example, but taking a look at the time, let me just skip to the next question. Is it possible to predict what drugs my repurposed drug has a high risk of interacting with? I guess you can in PharmaPendium, because PharmaPendium supports prediction of the DDI risk through a static DDI risk calculator. And basically what it does, it predicts potential metabolic drug-drug interactions between proprietary drugs and a panel of marketed drugs. And in this case, the proprietary drugs can also be your drug that you're currently repurposing. And importantly, the marketed drugs, PharmaPendium has a very extensive database of let's say, drug metabolic information on marketed drugs. So here we have the 238 unique drugs assessed as victim and 521 drugs assessed as perpetrator. I think this really sets PharmaPendium DDI risk calculator apart from competitors. And to do a calculation on your drug as a victim of a DDI, that basically the only thing you would need to determine in the lab is the FME value, which we just discussed, and for which enzymes, basically the combination of the enzyme and the FME value. And the rest of the data is drawn from PharmaPendium and gives you an overview of all the categories where there is a high risk of drug-drug interactions. Now, we don't really have time to go into too much detail around how this actually works and really show you a workflow, but this is typical output from the PharmaPendium drug-drug interaction risk calculator. And what we can see here highlighted in red, which indicates high risk, that, for example, Tofacitinib has a high risk of interacting with antiviral drugs. So in this case, if you put patients on antiviral drugs and combine that with Tofacitinib, where you try to suppress that cytokine storm as well, things can go really bad. And this is, of course, crucial information that you would like to have before setting up clinical trials with patients with COVID-19, where you really would like to then exclude patients that might be on antiviral therapy as well. Now, what we're doing, we're taking this one step further. We're working with the 7 pharma partners to further develop this PharmaPendium drug-drug interaction risk calculator. We have a partnership including Sanofi, Pierre Fabre, Elsevier, Boehringer Ingelheim, and 3 more partners. And basically, what we have done, we have improved the ease of use. Now what we just looked at was an Excel result. We've included this now into the product as well. We have improved the connectivity and have included also in-product working environments. And hopefully these improvements have then been validated together with the partners that will further increase the effective prediction and identification of potential harmful DDI risk. Now, we're currently aiming for releasing this new DDIRC in November. So if that's of interest to you and you are a DMPK professional for which predicting DDI risk is crucial, then this is definitely something that we would like to learn about and further discuss with you. Now, to kind of round this up, what we've shown you today is that PharmaPendium delivers critically extracted pharmacokinetic data on approved drugs that can inform dosing and dosing strategies during repurposing, that the extracted metabolizing enzyme and transporter data can be used to support identification of harmful DDI risk, and that DDI risk calculation in line with FDA guidance can be done through PharmaPendium early during development, because this is a static risk calculator. And in conclusion, access to critical data to support decision-making for repurposing of approved drugs. Keeping an eye on the time, I think we're 1.5 minutes over. If there's any questions, always feel free. We'll get back to you with the questions that you post in the question box already by email. And if there's any further questions, always feel free to reach out to us via email or LinkedIn. With that, I'd like to thank you for your attention and hopefully see you again for the next webinar. Thank you and bye-bye.
Thank you, Marnix. Bye.